Hi Bio30. Today we're looking at the very cool thing about your DNA and how it is the recipe for making all of the parts of your cells and your body and all the cell products. So we're looking at first of all what an amino acid is, which is the parts of protein. So there's 20 amino acids that in different organizing them in different ways makes different products. So just like we have 26 alphabets and can make endless numbers of words, with these 20 amino acids we can make many different proteins that are the structural parts of cells and therefore organs, as well as cell products like hormones and enzymes. So this guy here, Frederick Sanger, figured out that the amino acids in certain organizing them in certain ways with using different ones in these molecules will then end up being certain substance like insulin, which is a protein, or collagen or hemoglobin. Those are all proteins made of amino acids. So you have this diagram, which is basically all of the story of 18.2 all-in-one diagram. And we're looking at, when we look at the nucleus here of a cell, this is the cell cytoplasm here, the inside of the cell. This is the nuclear membrane. Our DNA is found in the nucleus, and what happens when we need to make some product, like let's say it's insulin in this cell, let's say it's a cell in your pancreas that's going to make insulin, the part of the DNA, that the chromosome that carries the code or the recipe for making insulin will open, and that code will be exposed, and then the code will be copied. Now, a scribe in olden times was someone who copied what the king would say. So think about that, the scribe, when you see the word transcription, because it is where a copy of the DNA code is done. So this messenger RNA is the guy that will come and copy the code, and that copy is taken out of a pore in the nucleus, and that copy is taken out into the cytoplasm. And the DNA then will coil back up, and now that copy is going to be read. And I often say you don't copy a recipe and with intention of making one cookie. While you have all the stuff out and you've got the recipe there, you will make dozens of cookies. When this code comes out to the cytoplasm, we will make many, for example, insulin molecules by reading that code. And where this happens is on the ribosome. Now, you guys have heard of ribosomes being like speckled all over through the cell, and maybe speckled on the ER to make it rough ER. What the the ribosomes are for is those ribosomes are actually workbenches for making proteins. So here's a ribosome that's basically split in half, and it'll clamp together and grab that RNA so that protein synthesis can happen, so the reading of the code can happen. Now, the DNA, remember, was a code. There's two sides, right? So let's say this side said CTGATA. Your RNA, remember, has no thymines. So when that code is copied, first of all, the one that's going to be copied, so it's this top one here, would be the sense strand. So only one of them is ever copied, the one that will that will make sense when the product is made. And when the RNA copy is made, it will be the complementary bases. So this side will be G. Remember that T binds to A. The G will be a C. The A, remember, has to be a U because RNA does not have thymines, it has uracil. T will be A will be U. So, for example, these are the sequences that I just copied from the DNA that was exposed to show the code for the product that's going to be formed. Now, this messenger RNA, I'll just change it to orange here, is going to be grabbed by a ribosome. Here's a nice big ribosome here. And interestingly, a ribosome is made of another kind of RNA called ribosomal RNA. So RNA is in three places. Ribosomes are made up of ribosomal RNA. Messenger RNA is the copied code from the DNA. And there's one other one, and that will be the transfer RNA, which we'll talk about in a second. So we're at the point where this messenger RNA came out. It's got the message of the secret code that it copied from the DNA. The ribosome has clamped it up and grabbed it, and it'll be grabbed by many ribosomes, and each one at each spot. Then the translation process, which is translating the code into a product, will happen at each ribosome. So over here, when we copied, it was called transcription. Over here, when we are translating the code and reading it to form a product of a certain protein, that's called translation. Now our ribosome is grabs up this messenger RNA, and whatever code that's on there. So remember I said that we had a GAC and a UAU that was on our messenger RNA. What's going to happen is that other guy, transfer RNA, it's kind of looks like a T shape. The transfer RNA will actually have three little bases on it, and there's many transfer RNAs, and the one that will match to the messenger RNA, which are called codons because they are the copy of the secret code, whichever transfer RNA has the correct anticodon that will be a complement will attach to it. So if GAC is here on my messenger RNA codon, then the anticodon that will match, remember this is still RNA, will be C, U, and G that will match exactly to G, A, and C. So if I write that on the side, my messenger RNA was a G, A, and C, and what would match that exactly is the complementary bases of C binding to G. Remember, it's RNA, so it has to be U that binds to A, and a G that binds to the C. So that's what's happening right here. And when the anticodon binds to the codon, what is on the other end of this transfer RNA is an amino acid. And that amino acid will be put as a structural part of the protein that's being assembled. Now, this slides along once the transfer RNA drops off its protein. And the next thing that happens is the next amino acid that will have the match to UAU is going to be the next one who joins in. So to match UAU, it will have to be a transfer RNA that has an AUA, and its corresponding amino acid will also join in. What's going to happen is after the entire code is read, the transfer RNAs will keep bringing amino acids until the code is completely read. And that string of amino acids is the protein that was just assembled. So in this case, I was talking about it being, for example, an insulin molecule. if you guys followed that a little bit, then you will be being refreshed with all the information coming on the pages to come, because it is basically step by step of this process of number one, DNA being read, that's transcription, and the messenger RNA copy being formed. Number two, translation, where that code, the messenger RNA codon, lines up on a ribosome, and the transfer RNAs match up to the exposed codons. And because the transfer RNAs carry amino acids, we're going to join up the whole string of amino acids according to the code, and there we will make our insulin. Coming up, we're going to see how we use the chart that's on the last page of your Chapter 18 booklet, because each set of 3 bases on messenger RNA actually code for 1 amino acid. So the next step is how we're going to look at what these actually code for. So GAC, for example, I'll just pop ahead. When you look at this chart, the first base being G, and the second base here, it tells you second base is a U, and the third base is a C, we can easily find what GUC codes for, and it is valine. So using this chart, we can see that GAC, pardon me. GAC is actually over here. It is aspartate. Sorry, I forgot from one slide to the next what we're looking for. Okay, so GAC actually codes for aspartate. UAU, we'd start over here and say that we're going to find UAU over here, and UAU is tyrosine, and remember the first base tells you that it's in this section, and second base being A tells you it's in this section. So UAU is tyrosine, and so when we go through the process, you just have to know that transcription is the copying, translating is the formation of the string of amino acids, and by using your chart, we can tell what the amino acids will be for every set of 3 bases on the messenger RNA called codons. So I put this into my slide here, and you can just review what we just said, is that here, transcription, remember the scribe copies. Transcription is when the RNA is making a copy of whatever gene is exposed here, which is the recipe for one protein. So transcription is when the messenger RNA is forming here. And basically, if you have a very valuable recipe locked away somewhere, like we have in the nucleus, we want to keep it there. So that's why we don't have the DNA coming out into the cytoplasm to where the factory is assembling. It is going to stay in the nucleus and protected by the nucleus, so by the nuclear membrane. So that's why we go in and make a copy. So if you have a special cookbook, you might go make a copy of one of the recipes rather than having the cookbook out where there may be chocolate batter spattering your cookbook. So messenger RNA brings that out to the cytoplasm, and it lands on a ribosome. Every ribosome it lands on, translation will happen, and you'll make many copies of whatever that code's for. But it lands here on the ribosome, and translation is where the code that's on the messenger RNA codon will be translated into a product, a string of amino acids. And that string of amino acids can be called a peptide chain. If it's really long, it's called a protein. Now, our little, whatever codons are exposed here, our transfer RNAs will land on there and match up to, for these are both RNA, so A's and U's will match up, and C's and G's will match up, and the transfer RNAs they transfer in an amino acid until you have the whole code read and the string of amino acids formed. Always the first one is going to be AUG because first of all, this chart tells you what the messenger codons are, so don't ever use this chart for DNA. Do not use this chart for transfer RNA. This chart is only showing you what the messenger RNA codons will code for. So AUG, it says here, is always the initiator, so it'll be the start of a strand, and we know when to chop off or end that strand, and that the protein is complete, because there are three terminator codons that will signal that this is the end of the strand. So if you see a strand that starts with AUG, you know that's the start. If it ends with UAA, UAG, or UGA, it will cut off there and finish. Now, when we go back to what we learned in our last topic, this word replication, that's just when DNA opens with helicase cutting it open and it makes a copy of itself, and that's what it does before it does any cell division. It will do a replication and make an X shape. So replication is a chromosome turning into a replicated chromosome. So don't get that word confused with any of the other two processes. Transcription is when messenger RNA forms a copy, a single-stranded copy of the code that codes for that particular protein for a gene, for, let's say, insulin. And translation is when the code is assembled, the code is read by and assembled by the transfer RNA, bringing in the correct amino acids, and the big giant strand of amino acids will lump up and become that functional protein. Okay, this is the chart that just says DNA is that double helix, that twisted ladder that you have seen many times. Messenger RNA will be the one that makes a copy of the DNA but makes a single strand. The transfer RNA by the way, messenger RNA is the codon. Transfer RNA is the anticodon. It's going to have the complementary bases that will attach to the messenger RNA and carry some particular amino acid. And ribosomal RNA is just the ribosome itself. So remember, it's kind of a flower shape. That's the ribosome itself is made of ribosomal RNA. So there's our three kinds of RNA. And in your definitions, it just repeats the same thing again, that the codon is your messenger RNA sets of three bases. There'll be a start and a stop codon that will signal the beginning and the end of that strand of amino acids. The anticodon is the transfer RNA set of three bases. And this word, just like polymerase before, when we heard DNA polymerase brings bases in to make the other side of DNA when we replicate, RNA polymerase is a similar kind of thing. It basically helps the copying happen in the nucleus. When messenger RNA is copying the DNA, RNA polymerase is helping to assemble the correct complementary bases. So polymerase is always like bringing the right base into place. Except this one means that it's bringing the right base into place to make messenger RNA. Okay, so transcription, if you can see on RNA polymerase is this big pink blob. It's helping the correct bases form this messenger RNA strand, which is full of codons. And you can see that the one being copied here is this one at the bottom. This is the blue one here is being copied, and we're forming this green strand of messenger RNA. So the one that we're copying at the bottom would be called the sense strand, the one that's being copied, and it's the one at the bottom. And the one that's never copied is the antisense strand. So we are exposing the sequence of DNA so that messenger RNA can form the complementary matching base pairs. And that is what transcription is, copying the exposed base pairs. Okay, remembering that we will always have uracils going in there, and there is no thymine in RNA. So if you look at your DNA sample right here, CAA, for example, you know that your messenger RNA will be a G and a UU because there are no thymines. If DNA has a TGG, your messenger RNA will have the ACC as a match. So you guys will have to do this kind of work where you take a look at always, my tip is that you always label your strand. So if it tells you it's DNA, label it so you know that what you've got here is DNA. And it says what's the messenger RNA that will be formed. So you can write down the messenger RNA that will be formed is, put that label on the side, split your bases into threes because that's how your chart works. And then you can start doing this transcription where you match them, remembering that T bonds to A has to be a U, G bonds to C. And remember that if you can remember that your messenger RNA has uracils, this will be easy. But again, the practice here is what's going to make it really quick to do, is you practicing how to make these complementary bases. And so keep doing that. Try to make the complementary bases. This will be a very important thing to do. Now, what happens with those then is you can go into your chart, and AUG is for methionine or start codon. So if you take a look at that code, A is your first one, U is your second one, AUG, so it's found really quickly here, is methionine, and methionine is a start codon. Next one is, UCC, you can check that out. It codes for serine. GAA, you can check it out, codes for glutamine. UCU codes for serine, and UAA is one of the stop codons. So that's about like as hard as it's going to get for this chapter, being able to write down, but you do have to practice this so you're good at it. Make the complementary bases of the DNA so you can make your messenger RNA strand, and use the messenger RNA at the top of your chart. It does say that this is only for messenger RNA, so that is the only bases that you can use this chart for, is messenger RNA. And repeating then, translation is when we go to the nucleus, out of the nucleus and into the cytoplasm, and that strand is grabbed by a ribosome, which is our workbench, and the complementary bases will match up. So if your messenger RNA is GUA, your transfer RNA CAU will match up to it. It'll carry a particular amino acid and attach that to our string. Now, this, by the way, is not the one that would match over here. UAAA would be matched by a UUU, and that would come in, stick on, and then the next amino acid would be added to our chain. So there is an error there in the book. Okay, and so you just have to remember that your messenger RNA sets of 3s are called codons, and your transfer RNAs are called anticodons. Okay, chart and review pictures, and we can try another question like this if you'd like, and as long as you guys, I think that's the same question actually as before, try those so you know how to do them. See, oh no, it is a different one. Sorry. So this is, again, the way you do this is, I thought it was the same one, but it's a different one. Okay, so label your strand. It tells you that's DNA. You want to make the RNA copy here. This one, again, is going to be AUG, which I know is, like before, methionine or start codon. And the next 3, CGA. Put your matching bases in there, check your chart, and that should be alanine. And you guys can make sure that you have the correct, you can try it for yourself, amino acids, the correct amino acids in the end. So they should be methionine, alanine, lysine, histine, and then the stop codon. So check that you can get the correct list of amino acids. And remember that you're going to go to the chart every time with your sets of 3. Okay, this last little bit on proteins just shows you that an amino acid is always got this amino group with the nitrogen. It's got a single central carbon that's attached to the R group, which means remaining portion of this amino acid is different for all 20. They all have a different R group. And there's a COOH carboxyl on it as well. So if you are looking at how amino acids join together, remember this concept from Bio 20. You've got to remove a water, an H and an OH from 2 amino acids so that they have a spot to join together here. And so here is where you have the NCC of one amino acid and the NCC of the other amino acid, and they'll bond together. Water will be left over. So it's a dehydration synthesis process. I put this in so that you can see, for example, in here where complete amino acids are. And remember, they're NCCs. So there's one complete amino acid. There's one complete NCC, NCC, and NCC. So there's actually 4 complete amino acids in this photograph. Just so you could see how the amino acids look when they're stranded together to make a protein. Okay, and the things that they're used for is things like the proteins in cell membranes, parts of cells, hair, nails, for structures of your body. Functionally, these proteins will be enzymes, hormones, antibodies, hemoglobin, and they're essentially made every day, many hormones, enzymes, many of these things formed every day just by reading the genetic code in your DNA. So that's a pretty cool thing. And the last slide is just talking about studying the genome is genomics, and studying the proteins that are formed is proteomics. Okay, so that wraps up how this is done. And I would like you to go do some practice that is listed in our homework to practice doing this. One thing that some of the questions may ask as well is, what is the transfer RNA that would match up to this DNA? And the main error that sometimes students have is that when they say, well, the messenger RNA, if it's AUG, my transfer RNA must have had an AUAC. For this one, GCU, my transfer RNA that swung in with amino acid had a CGA. For this one, obviously it's AAG. So my transfer RNA would have been UUC. But what some students do, if they're not careful, is they take these anticodons and they go to the chart and they find amino acids that don't exist because the chart is only good for messenger RNA. So if you ever have to, you know, first of all, label what you're doing here, it says this is our DNA strand, asks you the messenger RNA strand, that's where you can go to your chart, is with the messenger RNA. They may ask you the transfer RNAs and what they were, what those anticodons were, but do not use that for your chart and finding amino acids, because that chart is only for messenger RNA. So that is my final tip as you go and do some practice. And thank you for joining me for 18.2. I will see you soon for mutations.